Great. Thanks a lot, Lisette. Hello, everybody. It's really an honor to receive this award and also to present some of the work we have done over the last few years here today. So very briefly, as Lisette already said, I'm from the University of Vienna. I'm the head here of the Neuromechanics Research Group, and my group focuses on 3 research lines. One is more the musculoskeletal modeling, the second one is mechanobiology, and the third one is more on motor control. The presentation here I prepared for today focus more on the middle part, so on the mechanobiology, but as you will see, it's also very much influenced by the musculoskeletal modeling part. All right, so we all have bones. Healthy bones facilitate movement and are essential for a healthy life. When we move, our muscles create some forces, and these forces create some loads on our bones. It's these loads which then shape the geometry of our bones. And if you have been at the ESMAC conference at the opening ceremony this year in Oslo, and you have heard the song Dry Bones, you know how many bones we have in our body. Unfortunately, many individuals, including children as well as adults, develop bony deformities, especially at the femur. So far, it is not known what mechanical loading situation leads to this deformity. These deformities can lead to an altered gait, in-toeing, as shown here in this video, but also to pain, to tripping, and they act as risk factors for clinical conditions like osteoarthritis. Surgeries, where the medical doctor cuts the bone, reorientates the bone, and screws them back together, are used to address these deformities. It is, however, not known if the surgery normalized the loading situation which led to the deformities and therefore prevent reoccurrence. My long-term vision is to shift the treatment paradigm away from these reactive surgeries to proactive interventions to prevent the development of these deformities. To achieve that, we need 2 things. First of all, we need a way for early detection of abnormal bone loads. And the second is we need non-surgical interventions to normalize these loads. My presentation here today focuses more on this first part. There are several fundamental steps we need for an early detection of abnormal loads. First of all, we have to understand the complex interplay between our movements, the internal loads, and bone growth. So our bone loads are shaped by our anatomy and the way how we walk, and all this is influences then growth. And second, we need a way to predict subject-specific bone growth, because only then we could identify high-risk individuals early and therefore enabling preventive load-modifying interventions. In these papers which I will present here, I will focus on 3 research questions. The first one is what factors influence in vivo bone loads? The second is how do in vivo bone loads influence bone growth? And the last one is, can we predict subject-specific bone growth? So I put together 6 publications. Most of them have been published over the last 2 years, and one is currently under review. Let's get started. In this first paper, we wanted to investigate how the femoral morphology, so especially the neck shaft angle, so the angle between the neck of the femur and the shaft, as well as the anteversion angle, the angle between the neck and the knee axis, if you look at it from the transverse plane, how this influences our joint loads. It has been already shown in a few studies that an increased anteversion leads to an increased hip joint load, but the interaction between these 2 very important anatomical features have not been investigated yet. So what we did is we used musculoskeletal models where we created 25 different models where we altered the anteversion angle as well as the neck shaft angle. So at the end, we had 25 models with the different combinations of this angle. We then used the motion capture data of 1 healthy participant and investigate how this changes the joint load. What we found here was that as expected, an increased anteversion angle led to an increase in the hip joint contact force, shown in the top figure, but also an increase in the knee joint contact force, especially at the second peak. But this is now only the influence of the anteversion angle. When we then also looked at the neck shaft angle, we could see that a decrease in neck shaft angle had a minor impact. However, increasing the neck shaft angle also had a huge impact on the hip joint loads. When we're looking at the knee joint loads, we saw that increasing as well as decreasing affected the hip joint loads. And this was caused by altering the moment arms of certain muscles, which then led to more co-contractions and led to this increased joint load. To put this into a clinical context, there are several studies which showed that an increased anteversion but not retroversion is associated with hip osteoarthritis. And this so our study might help to explain that, because we saw increased loads with an increased anteversion angle. Furthermore, studies showed high incidence of hip and knee pain in children with idiopathic increased anteversion, which also is in line with our findings. Another study showed that a neck shaft angle beyond 134 degrees increases the likelihood, the risk for knee osteoarthritis by a factor of 8. And we also saw with the increasing neck shaft angle a huge increase in the joint loads, which might help to explain this clinical observation. Importantly, what I want to point out here is that there is a huge variability in the neck shaft angle and the anteversion angle even in healthy participants. So even not only in children, but also in adults. So therefore, if you really want to quantify subject-specific joint loads, it's very important to account for the bony geometry. And this also taking in mind, because now more and more smartphone-based tools get developed where you can estimate more or less joint loads, but be aware of it, that these methods do not account for the subject-specific geometry, which will influence your joint load. A big limitation of this study was that we only used motion capture data of one healthy participant. And this limitation leads to the next study. In this next study, we wanted to investigate if either the femoral morphology, so the neck shaft and anteversion angle, or the subject-specific gait pattern has a bigger impact on joint loads in children with CP. Probably most of you know cerebral palsy, CP. It's caused by non-progressive damage to the motor cortex and or spinal cord. This can lead to altered muscle activation, muscle morphology, and then in the long run to bony deformities. And all this leads to walking disabilities, as we can see here in this video. And we wanted to know if now the asymmetric bony morphology or the asymmetric gait pattern is the main contributor to the asymmetric hip joint loads. How did we do that? We had motion capture data and MRI images of 12 children with CP and 15 healthy, typically developing children. We created personalized models taking into account the personalized femoral geometry. Then we used OpenSim to run simulations and calculate muscle forces and joint contact forces. And when we then compare the left with the right side, we get the real situation. So we see how the joint loads differ. But this can be due to different geometry between left and right and due to different gait patterns, so different asymmetric gait pattern. To address our research question, we conducted a very simple form of so-called what-if simulations. So we run additional simulations where we, on one hand, kept the morphology the same. So the geometry of the left and the right femur was exactly the same. So therefore, we could investigate what's the impact of the gait pattern on this asymmetric hip joint loads. And then we conducted additional simulations where we kept the bone subject-specific, so they are subject-specific for the right and the left leg, but we kept the gait pattern symmetric to investigate what's the impact of the morphology, of the asymmetric morphology on this hip load. What we found was that in our CP children, as well in our healthy, typically developing participants, the gait pattern, which is shown here in green, had a larger impact compared to the morphology. Furthermore, as expected, the asymmetric hip joint loads were a lot higher in the children with CP compared to our healthy participants. Summarizing, gait pattern had a really big impact on joint loads. We therefore believe, from our point of view, that clinical intervention should not only normalize the pathological geometry, but should also try to normalize the loading situation and therefore potentially the growth of the femur, of the bones. If this is possible with only physiotherapy or gait retraining, or if a surgical intervention is needed to change the gait pattern as well, was beyond the scope of this study. And the big limitation of this study was that we only looked at joint loads. So you can imagine you can have the same joint load, but if you have a growth plate which is at a different position or orientation, this may lead to different loadings on your growth plate. And it's actually the loadings in the growth plate which define how the femur, how the bone grows. In this next study, we tried to overcome this limitation. So this was still a study which I conducted when I was at KU Leuven. So again, we know already the femoral anteversion angle and the neck shaft angle, and usually in children with CP are born more or less the same as healthy participants, but in healthy participants, usually these angles decrease, and in some children with CP, they do not. And we wanted to know what are the reasons for the development of femoral deformities in children with CP. And the second research question was, why do not all children with CP develop these proximal femoral deformities? So again, we had 16 children with CP and 6 healthy children. We used the motion capture data to calculate subject-specific muscle forces and joint loads. I then also had finite MRI images of one participant and a finite element model, which I created in a very time-consuming way. So it took me 3 weeks to create this model for a previous publication. And then we applied the loadings from the musculoskeletal simulations to this finite element model to calculate the stresses in the growth plate, and then using a mechanobiological model to simulate growth. And then we could compare the final geometry with the original geometry to investigate how the geometry changed. So we did this for the healthy participant as well as the CP participant, and then we created 2 groups. So one group of CP children which had predicted growth values which were within 2 standard deviations of the healthy participants, and a second CP group where the predicted growth was outside this normal range of 2 standard deviations. And what we found in the CP group with predicted pathological growth, they had a less posterior oriented hip joint contact force, which was likely caused by the missing hip extension in terminal swing. Interestingly, in the CP group, which had predicted normal growth, their sagittal plane kinematics was similar to the other CP group. However, they used a different pelvic and hip movement strategy in the frontal plane, and this led to a normalization of the hip joint contact force vector to values which were within the normal values, and therefore to normal typical growth. So summarizing, I believe this workflow has the potential to pinpoint certain specific features, gait features, which might lead to deformities. The big limitation of this study was that the geometry of the final element model was not personalized. And this was because, as I mentioned before, it took me 3 weeks to create one of these models. And we also didn't have the medical images from these participants to create the personalized finite element model. As you probably by now can assume already, these limitations led to the next study. This study conducted the first out is Willy Koller, my PhD student, and we published last year. We had Again, 13 typical developing children and 12 children with CP, so altogether 50 females. And we created a semi-automatic workflow to create these finite element models. How does it work? So in these finite element models, you usually need a hexahedral mesh, which has to be aligned with the growth plate. And creating these meshes has been, in my postdoctoral period, very, very time consuming. So we now developed this workflow, which enables to create these models in approximately 1 hour. So again, compared to my previous study, where I used this workflow, it took me 3 weeks. The workflow is publicly available on the GitHub page, so we encourage peers to use it. Importantly, previous study who used this mechanobiological workflow, they were limited to a sample size of a maximum of 4 participants. We now had at the end 50 females, so a considerable larger sample size to these previous studies. So we used then this workflow, the one I explained already in the previous study, and we estimated the stresses in the growth plate and calculated the osteogenic index, which says that the red area is where growth is promoted and the blue areas is where growth is inhibited. And in our healthy participant, we found this ring shape, as you can see here. And these are examples for 3 healthy participants. So the shape was quite consistent between the participants. In our CP participant, we found a different shape, and the most prominent growth was on the lateral side, as shown here in this video, on this figure. And if you look at 3 participants, you can see there was quite large differences between participants. Some had more a shape which was similar to our healthy participants, some had very different ones. And as expected, the variability in the shape and distribution of this osteogenic index was a lot larger in the CP compared to the healthy participants. So in conclusion, the ring shape which we found here in the osteogenic index is in agreement with experimental observations, which actually shows that the growth plate changes during growth its shape from a convex shape to a more flattened shape, which is in agreement with the ring shape which we saw here on these simulations. We found that many, but not all children with CP have different osteogenic index. And as highlighted before, this workflow allows to create these models in a very easy and fast way, which only takes approximately 1 hour. The big limitation in this study was that we only simulated growth at the proximal growth plate, but the femur also has a distal growth plate as well, where also growth occurs actually more than at the proximal. And this led then to the next study. So this study we published, I think 2 or 3 months ago. And in this study, we used the same data again and we extended our workflow. So as highlighted here, where on the left side, growth actually happens 70% at the distal growth plate. We then extended the growth plate tool, which we previously developed in the previous paper, which I showed you, to also include the growth plate at the distal growth plate. And then we used musculoskeletal simulations to not only calculate the knee joint contact force, but the medial and lateral knee joint contact force, as well as the patellofemoral contact force, to make it as realistic as possible. We then divided our CP group in different groups. In a CP group with a normal knee flexion angle, one with a low knee flexion angle, but there we only had 1 participant, so I'm not going to talk about these details at all, and 1 group with a high knee flexion angle. Why did we divide it in these groups? Because it's known that the knee flexion angle has a big impact on the knee joint contact forces, which of course will have a big impact on the loads in our growth plate. What we then found is that there were quite some differences in the distal growth, in the osteogenic index of the distal growth plate. Again, red areas, growth is promoted, blue, growth is inhibited. And as you can see from the top to the bottom, at the bottom were the CP children with the highest knee flexion, there we could see that growth was primarily promoted at the anterior region. And if you can imagine, if the bone only grows or more grows on the anterior region for a long period, this would lead to this anterior bending of the femoral shaft. So therefore, we then also analyzed our MRI images of these participants, and we indeed found a significantly higher anterior bending in the CB children with a higher knee flexion, which is kind of in agreement with our simulation result. So it seems like that the high knee flexion during the stance phase alters the stresses, the loading in the distal growth plate, and might lead to more promoted growth in the anterior region of the growth plate. It seems to be a plausible result because we also observed a higher anterior bending of the femoral shaft based on our MRI images, which we had from these participants. But again, we also still have limitations. In this case, we didn't have any longitudinal data to validate our growth simulations. And this leads now to the last study of what I want to present here today. So it's a study which we just submitted a few weeks ago, which is currently under review. So in this study, we collected some data, some longitudinal data over the last few years. And we had 10 typically developing healthy participants. And we collected MRI images and 3D motion capture data over a period of 2 years. On average, our participants were around 10 years old at the first data collection session. So we had the MRI images and the 3D gate data, so marker data and the ground reaction forces, which we could then use for our multi-scale modeling workflow to predict growth. We could then investigate if our growth predictions agree with the measured growth from our longitudinal medical images. Again, we use the same workflow as already shown before. We have the motion capture data and the MRI images. We have personalized musculoskeletal models to quantify the loads. And we also have a personalized finite element models, which accounts for the different position of the growth plate and orientation of the growth plate. We then quantify the osteogenic index, use the mechanobiological model to predict growth, and investigate how the geometry changes. So what we can see here is on the y-axis, the vertical axis, our predicted changes in the anteversion angle, and on the x-axis, the horizontal, the measured change in our anteversion angle. And what we found is when we use our multi-scale simulations only as an input, we can explain approximately 40% of the anteversion development. When we then add the age and the anteversion at the first data collection, as well as the hours of sport our participants did, we get a better prediction and can predict approximately 66% of the development of the deformities. So just to put this in context, additionally to measuring the data I mentioned before, we also gave our participants for at least a week during this 2-year period activity tracker so that we know approximately how much sport movement they do, and we also let them fill out a survey so that we know, okay, what kind of movement, how many hours of a week they participate in sport and so on. So we had this information as well. But again, these predictions are now solely based on on the data we collected at the first data collection session. So summarizing, our multi-scale simulation workflow takes into account the personalized femoral morphology, gait pattern, as well as the personalized muscle and joint contact forces, and it seems to be sensitive enough to identify and predict the development of the anteversion angle. Importantly, we also tried to predict the changes in geometry only based on the femoral morphology, only based on the gait pattern, or only based on the joint contact forces. And all the separate parts of this workflow by itself was not able to lead to any significant prediction. So it highlights the importance of taking all these different aspects into account. So going back to the first growth prediction study, which I showed you today, where I told you, okay, the hip joint contact force orientation can identify if you have pathological growth or not. This is actually not true. This was true if you have a model which is the same finite element model, if you would have exactly the same current geometry. But if you take the personalized geometry of your bone into account, so the shape as well as the orientation and position of your growth plate, this is not the count. You really have to take all these aspects together into account to get to realistic estimations of growth. So this was a first, let's call it a first pilot study, I would say, because we only had 10 participants in this study. So there's of course a need for larger sample sizes and also for including pathological participants which do not have a normal development. Sorry, this slide should not have been here. So taking everything together, I hope I could show you that musculoskeletal simulations can be used to estimate internal loads. Even if we know there are a lot of model parameters included, they still can lead to clinically useful information, which might help us to explain to explain certain clinical observations. The musculoskeletal geometry has a big impact on internal loads. Therefore, for people, trying to estimate joint loads on a subject-specific basis, that is very important. Then the subject-specific gait pattern is even more important than the personalized geometry. This is especially important for people doing finite element simulations. Many people then just use, for example, the data from the Charité, where you have people with instrumented implants, where you measure actually the loads. But then if you want to apply to a very different population, this might not really lead to realistic estimations. We developed the workflow to estimate the growth plate stresses at the proximal as well as the distal growth plate. And we did the first validation, which led to quite promising results. Future work, we of course have to increase the sample size. Then we want to investigate how these loads actually change over time. Is the growth plate stresses in our healthy participants constant over time, or does it actually change? And also develop a workflow how we can non-invasively change the loading at the growth plate and therefore potentially change the growth plane of the bone. So coming back to my vision, I think it would be really nice to move away from this current reactive invasive interventions, because currently we just wait until there is a deformity and then we address it in a surgical way, which is of course also very costly and not very nice for the patients. To move to these proactive non-surgical interventions where we could potentially detect abnormal bony loads before the deformity develops, and then use innovative, non-invasive or non-surgical interventions to normalize the loadings and potentially prevent the development of the deformities. At the end, I'd like just to thank and highlight my previous mentors throughout my career so far and my current collaboration partners, considering that this is a ESMAC event right now. Kind of, when I prepared this presentation, was quite interesting because most of the current collaboration partners I met at the ESMAC conference, which highlights the benefit of participating in conferences and being part of a scientific community. Last but not least, my team at the University of Vienna. It's not the whole team, but most of the participants, most of my team members. Quite exciting. 3 of my PhD students, Willy, Alex, and Bernhard, are very close of finishing their PhD, so they are very likely to finish within the next 6 months. So that's very exciting. Some pictures of our social activities. Summary of the papers again. And with that, I'd like to thank you very much for your attention.